We're good to go. So hi, everyone. Welcome to this package demo from Federico Agostinis about the NewWave package for dimension reduction and batch effect removal for single-cell RNA-seq data. And I'm sure you've heard the housekeeping items before, but just in case, if you have any questions, please put them in the Q&A tab. And I think we will have the question session after the presentation. You can also upvote other people's questions in the Q&A tab. And in case you would like to ask your question live, there is also a raise hand feature that we can bring you up on stage. I see you have already discovered the emojis to give feedback to the speaker. And finally, the recording of this session will be available just after it finishes in the schedule. So I think that's all the housekeeping items. So I will leave the word to Federico, who will take you through the workshop.
Thank you. Yes, I would prefer to answer the question at the end of the presentation. And now I will start. So we create these packages in order to overcome the gap that has been generated between the throughput of the experiment and the capability of analyzing this generated data. We started from zinbwave, that is an already existing R bioconductor package, and we implement mini batch stochastic gradient descent optimization and a framework that allow us to work with HDFI files. We decided to use a negative binomial model following the observation that droplet sequencing technologies do not induce zero inflation in the data. Thanks to this improvement and using a PSOCK cluster to massively parallelize the computation, we are able to speed up the computation while keeping the good performance that zinbwave already had. Those are the R bioconductor packages that we are going to use. And first of all, I will give a brief theoretical introduction to the model. And then I will make you see how to use NewWave with different input object and the best way to use it related to your hardware. So NewWave assumes a negative binomial distribution where the mean parameter is related to each single observation, while the dispersion parameter can be set equal for a gene or specific to each gene. I'm going to refer to this modality like gene-wise dispersion. And this is the relation between the mean parameter and the variable that we want to insert in the model. The X matrix represent the cell-related information that we have on our data. That those variable could be, for example, batch effect, and the beta matrix is the related parameter matrix. V matrix represent the genes level information like GC content, and gamma matrix represent the parameter matrix of B. W represents the low-dimensional representation of our data, and alpha is the matrix of the loading of this low-dimensional representation. NewWave is designated for estimates all the parameters, so beta, gamma, W, and alpha. But if you don't need to do batch effect remotion, you can just use W and alpha, and the X matrix and the V matrix will be set with only the intercept in order to normalize the data. This is the specification of the estimation of the dispersion parameter, and there are some steps used in the model. There is we start with an initialization step that is done separately for beta and gamma, that are our regression parameters. And for those, we will use a ridge regression for the initialization. For our low-dimensional representation, we will use a PCA just for initialize them. And then it will start the optimization. First of all, the optimization of the dispersion parameters that, if it's set genre-wise, can be done massively parallelized, and it is done using maximum likelihood estimation. Then there is the cell-related parameters optimization that could be massively parallelized, and the same for the optimization of the genes-related parameter. While the first step of this, while the initialization is done only one time in the model, the optimization will be done iteratively until the convergence of the algorithm, or until 100 iterations are reached. In order to reduce the memory consumption when we want to use NewWave with a big data set and with a large number of cores, we used a PSOCK cluster combined with a shared object R bioconductor packages. These packages allow us to share a single object between different cores without the increasing in memory consumption. And so there is a little increase in the memory consumption for each core that is not comparable with an increase needed for the duplication of our data. Thanks to all those improvements, we can have a huge benefit in terms of time consumption. And for reduce even more the time consumption, we can use some mini batch approaches that can be done separately for each of the optimization steps that you can see above. I'm going to show how to use NewWave with two different objects. First of all, with a single cell experiment that with a matrix stored inside, and then a single cell experiment with a delayed array stored inside. For the first example, I'm going to generate some data using Splatter. Splatter is an R bioconductor package designated to generate single-cell transcriptomic data with a high level of customization. In this implementation, I want to simulate a batch effect that we are going to correct using NewWave, and I will set some additional batch parameters in order to create a more complex batch effect. So I will load the packages, and then now I will simulate the data. As you can see, I will simulate 1000 cells, but we divided in two different batches of 500 cells. And then I will create 10 different groups of 100 cells. You can think you can think the group as a cell type. So our aim is to remove the batch effect in order to have a good clusterization of our data, and a differential expression probability of 0.2. Okay, so let's see how our data object is a single cell experiment with 1000 cells and 10,000 genes. We can see that there are some other information in the call data slot that we are going to see it later. It is a matrix, an array, so it is the first example. But for this workshop, I will use only the 500 most variable genes. I'm going to select that on this single cell experiment. Now we can see call data. Call data has a batch variable that represents our batch, and a group variable that represents our cell type label. So there is a very important consideration that we need NewWave needs to have the batch effect as a factor variable, so I will force it to be a factor. Before starting the sample, I want to see how our data are distributed in the space. So I will do a PCA and then a t-SNE in order to plot it. And as we can see, the color of the points represent the cell types, and the shape of the points represent the batch. There is a clear batch effect in our dataset, and this will give a bad result in terms of crystallization. It has an adjusted round index of 0.06. That is not so good. And for the PCA, I used 10 variable for our latent representation, and I will use 10 variable also for NewWave. So let's start with NewWave. I'm gonna show some different implementation. So some very basic advice. The verbose option has a default equal to false. And in this vignette, I will change it in true, just for explanatory intention. And I suggest you to do not do it for when you use a big data set, because it can sensibly slower the computation. And when we will see the mini batches, there are no concern about the dimension of mini batches, but I always use the 10% of the points, and I suggest you to do the same. This is first of all, we are going to see the basic implementation of NewWave. So let's see the parameter of the function. Data is our single cell experiment. In the X parameter, you set the name of the batch variable that is stored in the call data of the data. K is the number of the variable for your that you want for your latent representation. And verbose equal to true, I set it in order to see what is happening. If you need to set some genes related parameter, you can use the V parameter, and it has exactly the same management. You can see that there is a time of setup needed to build the cluster, even if it is done of only one core. Yes, sorry. For this basic implementation, I will use one core that is the default of the program. After the setup, there is an initialization. You cannot see very well, but when it will have finished, it will be okay. Now it's clear. And there is a time of initialization, and then it starts the optimization. Just like I said, it is done iteratively. Before the optimized dispersion optimization, the right optimization. So the right optimization means the cell related parameter, and then the left optimization, that is the left the genes related parameters. You can see the value of the log likelihood for each step and the time needed for each step. Also, you can see also the log likelihood for after the orthogonalization that is needed because the solution is not unique. If we want to create a faster model, we can set the children parameter, that is the parameter that manage the number of clusters that we are going to use for the estimation. So there is a bit higher time of setup, and this is normal because we are going to use 10 cores instead of 1. The time of setup is needed to create and share the object, and also for initialize the cluster. But after the higher time of setup, you can see that the optimization is really faster. So I will keep on the left this model, but on the right I will skip to the first model just to see the difference. And so you can see that we use the 20% of the time for each optimization, and think about that it is a small data set, so we do not see the magnitude of 10 cores used, but is a good result, and it could be also better with a large data set. If we want to see, so we use a even faster model, we can start to use mini batches. I will show you the two best way to use NewWave, but the first one best way is when you do not have a lot of cores, a lot of cores. So if you have just, I don't know, 2 cores, this is a good option. And so I will suggest you to keep the common wise dispersion, that is the default of the program. And yes, it is not written, but we will see that when we will use the GeneWise option, I will have to set that. So common wise dispersion and the mini batches for all the 3 steps of the optimization. So there are 3 parameters needed to set the mini batches, because there is 1 parameter for each step of the optimization. And gene disp represent the number of genes that we want to use in each mini batch for the dispersion estimation. N cell par means it represents the number of cells that we want to use in each mini batch for the cell-related parameters of optimization. And n gene par represents the number of genes that we want to use for the genes-related parameter estimation. And when we use the mini batches, the first iteration is still done using all our observation. We decided to make it work that way because we have seen that this brings to faster convergence. It's like setting a good basis for the mini batch optimization. In this case, we still have a lag time of setup that is an unwanted factor that we are going to eliminate. And then you can see that the first iteration is still, needs still a lot of time. I will show again the first on the right in order to see the difference. But from the second optimization on, we need it is very faster. It's 0.29 second against 5 seconds. Okay, and it needs also one iteration as one iteration below the first model. Then the very best model that we are going to use now is the one where you have a lot of cores that you can use. So in this case, we will use the generalized dispersion, and you can see that I set the common dispersion equal to false because the default is the common dispersion equal to true. And an important thing in this case, I suggest you do not use the ngdisp parameter. So do not use the dispersion parameter mini batch, because when we use generalized dispersion, we can parallelize the computation of the dispersion, and so we do not need to use the the mini batches for this situation, and it will lower the computation. So I suggest you do not use it. Let's see how it works. And again, for the first iteration, we always have all our data in the optimization, but from the second one, we will use only the mini batches. The time is our model is faster than the first, so for a very good, for better interpretation of the time, we will need more data. When you use the model and the command that you can see here, NewWave, you will have the latent representation stored in the reduced dim slot. So now I'm going to see if the latent representation that we obtained using NewWave is better or worse than the one of the starting data. So now I have 10 variables like the PCA, and I will use TSNI for visualization. So there is the batch effect has been removed, and there is also a good division between the cell types. So I'm going to see how k-means clusterization can identify those cell types, and the adjusted rand index has improved. Now it's 0.69. But if you want to see not only the latent representation, but you want to see all the parameters that we have described in the model definition, so the beta, gamma, W is the latent representation, but also alpha, and the dispersion parameter matrix, you can use NewFit, that it works exactly is exactly the same. It works exactly the same, but it applies again to a single cell experiment. But now the output of the model is not a single cell experiment. It is a list where it is a specific model, a specific object called the new model. And here you have all the metrics that we have speak about at the start of this workshop, and you can use it to check it, to see the weights. And there are also two functions that apply to a new fit output object, that is new IC and new BIC. You can see here there is the new fit output, and here we have our matrix, and with those we can see the IC and the BIC of our model. And this is a good way also for select the best model that you want to use, or the right number of latent variable. You can estimate various model and then benchmark them using those functions. Now I'm going to make you see how NewWave works with the layered array object and HDF5 files. So we are going to see how it works with out-of-memory data. As we know, there are some dataset that are very big, enormous, and so even use it, even charging it in our RAM, it could be very difficult, and the model, estimated model, is even more difficult. So it is the 10x brain data library that contains the 10x brain data. It is a dataset made by 1.3 million cells and 27,000 genes. And we are going to see that it is a single cell experiment. It is still a single cell experiment, so NewWave will fit good with it. And here is the dimension of our dataset. But despite the dimension of our dataset, its RAM size is really little. It's just 304 megabyte. And this is possible because it is a delayed array. So we are not going to use all the data in each computation. So NewWave use these packages that are called the delayed array that has some strength and some weaknesses. And the biggest strength is, of course, the RAM consumption reduction that we are going to see. It will be very big, but the cost of that is that at each time we read solo a piece of our dataset that is called a chunk, and it could be very slow, especially for a model like NewWave, because we estimate a model for each column and then each row, and then we do it more than one time iteratively. So if it's not done properly, it could be very time consuming. And for this reason, I strongly discourage to set variables equal to true, because we have seen that when we set variables equal to true, we have also log likelihood calculation at each step of the optimization. That is one of the most expensive task when applied to delayed array object. So it's better to set it equal to false. I just see the result at the end. And I suggest to use it only with big dataset, let's say more than 30,000 cells, because with a small dataset we will not have an improvement in terms of memory consumption, or sometimes we have also an higher time in memory consumption, and on the other hand we will have an increase in time consumption. So there are some difference between the implementation on the layered array and the implementation of the matrix. The first one is a small approximation in the initialization, and the second one is that we force the dispersion parameter to be generalized. And in this case, so the best way to use NewWave is with a dispersion of course, generalized dispersion, and a lot of course, and a mini batch on the genes related parameters and on the cell related parameters. I will not run this function because on delayed array is more time consuming, but it works exactly the same, and so you do not have to set some information about the delayed array. Now I'm going to show you some benchmark of NewWave between different implementation. This is the benchmark. Okay. You can see the figure with now you can see the figure, entire figure. Okay, I think yes. So As you can see, there are four different models, and there is NewWave applied to a matrix, NewWave applied to a delayed array object with 10 cores, and a NewWave applied to a delayed array object with 40 cores, and also a PCA. Yes, when applied to 10x brain data, we do not have the batch effect. So this time we have used the NewWave just for normalization and dimensional reduction. As you can see, the HDF5 implementation is the most time consuming with the same number of cores, but with the 40 number of cores, it is faster than our matrix implementation. So we have a reduction using 4 times more cores of the 50% of the time. And we are going to see also the RAM consumption result. So here you can see that the matrix implementation, but you do not see the x-axis. So we also before we have estimated various model from 10,000 cell to 1.3 million cell. So we can see that with a small data set, it's not really I do not suggest to use the HDF5 implementation, but when applied to big data set like 500, 600,000 or 1.3 million, we have an enormous decreasing in RAM consumption. About it's like the 10%. And even we use 40 cores, there is, yes, an increase in RAM consumption, obviously, but it is still the 20% of the RAM consumption of the matrix implementation. So in one hand, we have using 40 cores, using HDF implementation with 40 cores, we have an decreasing of the 15% of the time and a decrease of the 18% of the RAM consumption. So this is the strength of the delayed array implementation. You can find all those results and even some other results that compare some different implementation of NewWave, like the different parameters that we have seen, the mini batches, the common dispersion, the general-wise dispersion. You can see found all of those results on the application note that is in pre-publishing, and I hope it will be published soon. And thank you for your attention, and I have finished.
Thanks a lot, Federico. Are there any questions? I don't see anyone in the Q&A so far. We'll leave a little bit more time for people to think. Maybe I can ask a question in the meanwhile. I was wondering basically how sensitive the output, I guess, is to the, for example, the choice. So how do you choose K, for example? Does it matter? Is it important? Is 10 a good general choice, or do you try different values?
Yeah, okay. I think that the right number of variable is a tough task for every factor analysis, and I do not have the best number of K for NewWave. I suggest to compare it to the number of genes. And if you are undecided between some number, to just estimate different model and compare the result using HE and BHE. The only comparison that I have done, but is not here, is not also in the application note, is between 2 and 10 different 2 and 10 component. Yes, there is a big difference, and so 10 is a general number, 10, and I often use that.
Okay.
Yeah, so just to add a little bit of a detail, we have a function to compute the AIC and the BIC in the package. So you can potentially like run with multiple Ks and then compare. But of course it's going to be slow, right? Because you have to to estimate. It's not like PCA when if you do 50, then you can get the first 10 as for free. In this case, you have to estimate all the models.
Yes. So we have a question from Andrew. I will bring it up here. Is there a way to make an out-of-sample prediction of a cell coordinates using a fitted model?
You can, okay, yes, you have the weight of the low dimensional representation. So if you use new fit, you can pick up the alpha matrix and use that for transform your data in the low dimensional representation. You can do the same with the parameter of the batch effect or some other genes related variable.
So is it just a matter of doing like a matrix multiplication with one of the weight matrix, or do you have to like solve the optimization problem but fixing one half of the like the left matrix or the right matrix?
No, you have to do it by hand, like a matrix multiplication. There is no a function to do it.
So we have another question. I'll bring that up. How does NewWave compare to other batch effect removal tools for single-cell RNA-seq, like Harmony or Bbkn? So what is the main advantage of NewWave over these?
I think the qualitative performance are similar to zinbwave or sometimes even better. And so from the literature, we know that zinbwave has a good qualitative performance, and then NewWave use NewWave combine this good qualitative performance with an improve in the RAM consumption and time consumption.
Great. I guess another question I had, maybe I'm sorry if I missed if you touched upon this. So you initialize the coefficients using a ridge regression, I think you said.
Yes.
Is it the result sensitive to that? I mean, if you change a little bit, do you get very similar results if you change the initialization a little bit, or can you get actually quite different output if you change the initialization a little bit?
I never tried to. I tried to different. No, I do not try to change the ridge regression initialization, and so I don't know. It will be a good point for some future works to change the initialization, but for now I don't know.
Okay. But the convergence looks fine in general, or can you also have convergence issues?
I never know. For what I know, I tried to. I used for the application note. I used the NewWave on the 10x brain data and on another data set of a single cell that has 300,000 cell, and I never had with those implementation. I never had convergence issue. With some other that now no one can use because are not possible, and there was.
So just to be clear, it is possible to have like relative maxima, right, in the likelihood. So it is important how you implement, how you initialize. Yeah, so I don't know. It is an important point, and one that I don't think we fully know. Like, we don't really fully know the extent to which we go to the actual real maximum and not like a relative maximum.
Maybe there are multiple equally good.
Yeah, exactly. And it's probably very flat at the top, right? So
yeah,
who knows?
Yes, good. There's another question on whether you can use NewWave to integrate or remove batch effects for datasets from different species or different tissues.
It is theoretically possible, and has been used, for what I know, has been used also for trying to merge some bulk Rnaseq dataset with a single cell Rnaseq dataset. And in that case, it shows good performance. So yes, it is possible.
Great.
I wonder if we return again to Davika's first question here about advantages. So one advantage that comes to mind with NewWave is that it's really a statistical model, kind of end to end. Do you have any, are you thinking of any other ways you can kind of leverage the fact that you can really do inference on the parameters that are coming out of NewWave as opposed to some of the other methods which are really kind of just an algorithm? They don't permit any sort of inference.
Sorry, can you repeat the question, please?
So NewWave should let you actually do inference on like the model parameters that come out, like the left and right matrix, etc. Have you given any thought about what you could do with those quantities, like standard errors or confidence intervals on them, like how those could be used?
No, for now we have not worked on that, and that could be a future work topic.
Yeah, it seems like a potential advantage anyway.
Okay.
Yeah, I totally agree. Right now we don't actually estimate the standard errors for the betas and the alphas, but yeah, just for computational reasons, right? But we could, and that definitely would be something to explore. Yeah.
Great. I think maybe if there are no further questions, we will wrap up here. So since the next session starts already in 2 minutes, so people have a chance to to swap over. This was great. Thanks a lot, Federico.
Thank
Davide,
you.
Andrew,
Thank you very much.
Everyone who participated, and see you in the next session.
See you.
Thank you.